Hola, buenos días a todos y bueno, muchísimas gracias por asistir a creo que es la 27 edición de nuestro ciclo Marie Curie. Muchísimas gracias. Digamos que tenemos una norma en el instituto y es la de deciros brevemente una serie de normas de seguridad y que son muy sencillas, en realidad. Entonces, en el caso hipotético de escuchar una alarma, pues hay dos salidas de emergencia de este salón de actos. Uno es, pues, la entrada por la que habéis accedido al salón, y otra es esta puerta de aquí, que en el caso de que tengáis que hacer uso de ella, tenéis que girar a la izquierda, al fondo, y ya se sale. ¿Y dónde hay que ir en el caso de emergencia? Tanto si se sale por ahí como por aquí, al punto de encuentro que está en el jardín justo enfrente del edificio central. ¿Vale? Entonces, pues, en principio el mensaje ya está dado. ¿De acuerdo? Entonces, nada, voy a decir unas palabras de introducción a nuestro conferenciante invitado, que va a ser en inglés. Entonces vamos a cambiar a inglés. So, good morning, everybody. And I'm going to say a few words about our invited speaker, which is Professor David Jameson. So, just a few words. It is my great pleasure to introduce to our speaker, Professor David Jameson. Since I don't want to take up too much of your time, I will say just a few minutes about his scientific career. The academic journey of Professor Jameson began with a Bachelor Science in Chemistry from the Ohio State University, and then a PhD in Biochemistry at the University of Illinois. During his doctoral studies, he worked with very famous biochemist from Argentina, which is Gregorio Weber, which is also father of the fluorescent field in the 20th century. After his doctorate, Professor Jameson continues his career, first at the University of Paris, working with synchrotron radiation, which is very familiar to our work in the day, and then returned to the Professor Weber's lab. His career trajectory led him to the pharmacology department at the University of Texas Southwestern Medical Center as an assistant professor, before moving to the University of Hawaii, in particular to the Department of Biochemistry and Biophysics. And today, in this university, he holds a professorship in the Department of Cell and Molecular Biology. Professor Jameson research is mainly focused on protein-protein and protein-ligand interaction and dynamics, employing state-of-the-art fluorescent technologies. His current work involves key proteins such as dynamine and another couple of proteins with a name such as ARC 3.1, both in vivo and in vitro. In addition to his research contribution, he's very famous, very well known because of his book, which is Introduction to Fluorescent. And now we have this. Pilar has taken from the oven the second edition of this book. Please take this into account. Finally, I would like to remark that Professor Jameson has been one of the organizers of the International Weber Symposium, whose 12th edition will take place the next week in Genoa, Italy. Thank you, Davis, for joining us and give us this talk, which is a nano-history of fluorescence. Whenever you want.
Okay.
Go ahead.
Thank you. Thank you very much. Everybody can hear me? Okay. We even have one of the Weber meeting attendees in the audience who came to our Hawaii meeting. So, any rate, it's a pleasure to be here. I'm sorry my Spanish is not good enough to give this in Spanish. I do know one phrase in Spanish, estos no son mis zapatos, which is not very useful, I've discovered, but still, I use it. Any rate, I'm not sure how many people are familiar with fluorescence, so I'm going to start with a little background, but let me just say that fluorescence has developed into an extremely important technique in modern science, including especially in cell biology. And it's impossible to think of many cell biological things without doing fluorescence, and I'll explain why that is. But I thought it's interesting to look back at the history of this, and some of the history I learned here from Ulysses Acuna, who works here but is not here today. But I think it's, you know, as you get older, you start getting more interested in history, maybe because I've seen most of it by now. But any rate, so first, what is fluorescence? And fluorescence is the light emitted by an atom or molecule after absorption of electromagnetic energy. And it's been the developments of highly sophisticated fluorescent probe chemistries, new laser and microscope approaches, and molecular biology that has led to the novel applications we have now in the chemical, physical, and life sciences. And they're widely used for biophysics, biochemistry, clinical chemistry, and diagnostics. It's really, you know, the entire Human Genome Project, which you know of, was all done using fluorescence. But I'm not going to discuss that today. But why fluorescence? Well, first of all, it's pretty, but that's not a totally silly answer. When I first joined the lab of Gregorio Weber, the pioneer of modern fluorescence, he explained to me this was in the 1940s, before they had phototubes. Everything was done with the eye. He said it attracted him because he was amazed that by looking at changes in color and intensity, you can understand what's happening at the molecular level. And that's one thing that you still today, I tell people, if it's visible fluorescence, look at it. You know, you can gain information from it. It also provides information on the molecular environment. Fluorescent probes have been developed that respond to almost anything, electric fields, ions, magnesium, calcium, anything, pH, polarity, temperature, viscosity. There's hundreds and hundreds and hundreds of probes now. You can virtually design a probe. You want to study this process, you design a probe that can do it. But it also provides information on the nanosecond time scale, okay, because the fluorescent lifetime, it gets excited and it stays for nanoseconds before it emits light. So basically, fluorescent probes are essentially molecular stopwatches. You can attach them to a protein or a membrane, and you can see events that happen during that lifetime. So many times when you're planning an experiment, you want to know the lifetime of your probe is relevant to what you're going to study. So these are things you look up. But it's really wonderful. In my thesis, I relied on the dynamic aspects. And coupled with modern fluorescence microscopy, which is confocal and multiphoton, everybody uses 2 and 3 photon measurements now, and fluorescent proteins, which we'll talk about, like green fluorescent protein, it provides extremely detailed spatial information in living cells, as well as information on the dynamics of cellular components. It's also very, very, very sensitive. Working with sub-nanomolar concentrations is routine, and even single molecule studies are possible with some efforts. Experimental systems accessible to fluorescence are proteins. I actually think this is from one of Pilar's paper, this figure. From proteins, from cell organization and function, engineered surfaces for high throughput drug discovery, and even in live animals, people are doing fluorescence. But let's talk about the discovery and characterization. And let me first say I started this because I was giving a workshop 20 years ago in the Netherlands, and I said to the students, when do you think fluorescence was discovered? And they discussed, they said, probably about 1950. I said, no, it goes a little older. It was first mentioned by Nicolas Menardus in 1565, a Spanish physician and botanist who wrote on the medicines of the New World. He translated, well, he wrote from the documents that the Spanish in the New World sent him, and they were at this university, I think. Ulysses Acuna told me he went to these documents, actually. And this figure is from Ulysses Acuna, who went and looked at all of this early stuff. And he pointed out, that Menardus pointed out that the Aztecs would actually make cups from a wood. They'd fill it with water, and then they would get a blue tinge to it. And they would drink from this, and this was good for kidney problems, okay? And Bernardino de Sahagún, a Franciscan missionary, actually went there. Menardus only looked at the document sent, but he went there, and he said that the Aztecs called it coatl. And again, Ulysses Acuna wrote a wonderful article on the history of this, so you should ask him someday if you're interested. And there he is in Pilar, and also Ulysses gave me, in fact, he just gave me another piece of the kidney wood just now from Pilar. But this is a piece of the kidney wood which I got from Ulysses, put into an alkaline solution and then with a UV hand lamp. And instantly, this brilliant blue fluorescence starts coming out of it. It's just amazing. You can sort of see the piece of wood there. But it's just a stunning visual demonstration, which would be good for you people to set up in a class someday. But an early translation in 1574 by the Flemish botanist Charles de Lecluse gave the name of Lignum nephriticum, kidney wood, because it was useful for people with kidney ailments. In fact, it was very popular in the 15 and 1600s in Europe because of its medicinal values in treating kidney ailments. But actually, Robert Boyle, the famous of Boyle's Law, and many of you know of him, was inspired in 1670 by Menardus's report and investigated this system more fully. He discovered that with the wood, after many infusions, the wood lost the power to give color to the water. And he concluded there was some essential salt in the wood, which I thought was very clever, that's responsible. Then he discovered if you add acid, it went away, and you add alkaline, it came back. And this is from his actual document, which I thank Katie Reinhardt for giving me the original document. And he said how when he added a little spirit of vinegar, the curious color immediately vanished. But then he said when he added a few drops of oil of tartar del deliquium, it took me half a day to figure out that was potassium carbonate. That's what they called it. It would all come back again. So actually, he was the first to use fluorescence as a pH indicator, which is a very popular application today. And he really did use it as a pH indicator. In his work, he would use the lignum nephriticum to see if things were getting more alkaline or acid in his research. So he was doing this in 1670. In the ensuing centuries, the botanical identity was lost until 1915. William Safford sorted it out, and he wrote a wonderful article about this. And this is a picture from his book of a cup made from lignum nephriticum wood, and you can see the blue tinge of the water there. The identification of the fluorescent molecules only happened in 2009, right here, by Ulysses Acuna and his group. And it was very complex chemistry. They had to sort out the pathway that leads to a rearrangement, an alkaline-induced rearrangement, and finally gets you to this metalin, the fluorescent molecule. So that's the first fluorescent molecule, and the final identification was done right here in Madrid. So you should be happy of that. And those are the spectral properties up above it. Now, going to other people who contributed, David Brewster in 1833 discovered when a beam of white light goes through an alkaline solution of leaves, you can see a red beam from the side. That, of course, was chlorophyll fluorescence they're looking at. And chlorophyll fluorescence is now very important in studies on plant health and photosynthesis. And there's portable fluorescence instruments taken out into the field. But not only that, they also monitor this from satellites. And there's all satellite mapping looking at chlorophyll fluorescence from sending the lasers down from satellites. And you can see in areas in Africa and South America where you have a lot of the fluorescence. And this is all done from looking at the chlorophorescence, which I think is amazing. And we'll talk about another NASA thing later on. John Herschel was a mathematician, astronomer, chemist, experimental photographer, and inventor. He was an amazing person. He named the 7 moons of Saturn, and he named 4 of the moons of Uranus, which was discovered by his father, Sir William Herschel. There's a picture he took himself in 1867. He also translated the Iliad of Homer and invented blueprints. Now, when I was giving my graduate course in fluorescence in Hawaii last year, I realized none of the students knew what a blueprint is. Probably the students here don't know either. But this was the earliest way to do photocopying, essentially, like Xeroxing. And now, of course, everybody downloads a PDF. Who cares? But this was a photochemistry, and that's how people made photocopies back in the 1700s because of Herschel. But he made the first observations on quinine sulfate, and we're going to talk about how important quinine was to the fluorescence field. And he termed this phenomena epipolyc dispersion. And he actually wrote, this is from his paper, on a case of superficial color presented by homogeneous liquid internally colorless. That's his paper. And he described it as an extremely vivid and beautiful celestial blue color. And I loved, and you'll see again how they described things in those days. It was wonderful. It was so vivid, the writing. Now, quinine. This may be the most important molecule in the development of the fluorescence field. Quinine is a bitter compound that comes from the bark of the cinchona tree. And it's mainly found in South America, Central America, the Caribbean, and Africa. But it turns out when the Spanish came to the New World, they discovered that natives would treat malaria with the bark of the cinchona tree. And of course, there were malaria outbreaks in Europe too, or on the Mediterranean. So already as early as 1636, they were sending cinchona bark. The Jesuit missionaries from Peru sent them back to the Vatican and the New World and all around. But the actual antimalarial drug was not available until 1820 when Caventou and Pelletier in France isolated quinine from the bark, and you can see a statue of them in Paris because of this. Now, gin and tonic, if you ever have a gin and tonic and have a UV hand lamp, this is what you'd see, because gin and tonic water has quinine in it, of course, and that was the original gin and tonic. But how did it become a tonic? The cocktail was introduced in India when malaria was a persistent problem by the armies of the British East India Company. So they would make people drink the quinine in tonic water, but it was a bitter taste. So the British officers in the 1800s started adding a mixture of water, sugar, lime, and gin to make it more palatable, and that was the birth of the gin and tonic, just to get people to ingest the quinine. And it really is quite remarkable, actually, to see. Now, the real father of fluorescence, I think, has to be George Gabriel Stokes. Many people are familiar with him from Stokes' things on hydrodynamics. But he wrote this amazing paper, 100 pages long, and anybody that wants to get a PDF of this, email me and I can send you his paper. It's amazing to read, where he talks about this beautiful celestial blue color that Herschel had found, and he studies this, and he would take samples of quinine out. He used every light source available. He went out on a stormy night and held it up to lightning to see if he could get the fluorescence. And he did an amazing study, and he was the first to realize that there's some absorption of light and then re-emission of light. But in this work, he initially used the term dispersive reflection to describe the phenomena presented by the quinine sulfate. Fortunately for all of us here today, on a footnote on page 17 of the manuscript, he wrote the following: I confess I do not like this term, dispersive reflection. I'm almost inclined to coin a word and call the appearance fluorescence from fluorspar, a mineral, as the analogous term opalescence is derived from the name of a mineral. So if not for him, we would be having the nano-history of dispersive reflection. And I would have the second introduction to. So thank goodness he named it. And this is fluorspar. I had a piece at home, and this is on my desk, and I shine the light on it, and you can see why he was inspired to call it fluorescence. But he used a prism to disperse the solar spectrum and illuminate a solution of quinine. And he noted that there was no effect until the solution was placed in the ultraviolet region of the spectrum. So we'd move it through the spectrum. When it got to the ultraviolet, which he couldn't see, there he saw it. And then he wrote the most wonderful description of this. He wrote, "It was certainly a curious sight to see the tube instantaneously lighted up when plunged into the invisible rays. It was literally darkness visible. Altogether, the phenomena had something of an unearthly appearance." You have to admire the way he described this thing back then. And these observations led Stokes to proclaim that fluorescence is of longer wavelength than the exciting light, which leads to this displacement being called the Stokes shift. So that was all due to Stokes, who discovered that. But then we should turn to William Henry Perkin, who at the age of 18 got the idea of making quinine from coal tar. Well, all they had was a molecular formula, C20H24N2O2, and it seemed reasonable. They didn't know the benzene rings or anything. And he started to work out, and this was very important for the fluorescence history, as we're going to see, when he was only 18. He was also an amateur photographer. That's his picture he took. So he accidentally produced a synthetic dye mauve, which was a derivative of coal tar, and that's the color, okay? Fortunately for him, Queen Victoria loved this color, and she made a dress of it for the wedding of her daughter, so it became instantly popular. And then everybody, and Perkins recognized how important it could be financially to make dyes. He started a company to produce dyes, and the next produced a green and violet. Soon, they say the canal outside his factory turned a different color every week, depending on what he was working on. And histologists started using the dyes to stain samples within a decade of this. Unfortunately, the British didn't have a good patent system, and then this was really taken over, and they have monuments to him in London, how important Perkin, and he became very famous. There's the Perkin Award in chemistry, and all because of that. But Adolf von Baeyer, a German chemist, then started to work on this, and the Germans started synthesizing dyes, and they patented everything. And he synthesized this compound. And I guess I don't have to tell you what that was. Okay, maybe I do. That was fluorescein, the most popular dye in the world today. And I started wondering, why did he name this fluorescein? That's his paper. And in his original article in German, you can discover that he named it Flo, F-L-U-O, from fluorescence. But racine was from the German for resorcinol, because he reacted phthalic anhydride with resorcinol to get the fluorescein. So the name tells you the synthetic route, which I thought was very clever. And he got Nobel Prize in chemistry for all his works in organic chemistry. One of the first uses of fluorescein was in 1877. Well, I'm pretty sure before that it made the party circuit in people's houses. Oh, look at this funny color. But people used it for major groundwater tracing experiments. And Knopp put 10 kilograms of fluorescein in the Danube on Tuesday, October 9th at 1877. I read the paper. And about 4 o'clock at 4 in the evening, and then he translated the same effect, the effect of the same in the spring of the Ak was detected on the 12th. That was 60 hours later, which showed that this proved that the Danube and Rhine rivers were connected by underground streams. So this is the first water tracing things, and they still do it today, okay? Now, some people say, well, gosh, 10 kilograms of fluorescein, you couldn't do that today. Not true. You can, and in Chicago, where they started fluorescein, they put in 40 pounds of fluorescein on St. Patrick's Day to turn the river green. And actually, they used fluorescein for years. Now they use proprietary vegetable dye, okay? But they use that. Oh, and here they are adding the fluorescein. You can see them dumping it out of the back of the boat on the Chicago River.
pensais que ça allait être des zones, mais pédale.
This happens every March 17th. So
C'est vraiment du vrai produit chimique.
so much for the Environmental Protection Agency. But they put it in the beer too. You could drink fluorescent beer in Chicago on that day. But in 1887, Ceresol started to make rhodamine, which is the other really important early probe, and it gives this beautiful red color. And he worked for von Bayer and worked for the company BASF in Germany. But these things were brilliantly fluorescent, and he named them rhodamines from the Greek rhodon for rose, and then amine since they were basic. So again, the name tells you something about the molecule. And he developed all the rhodamines we use today back in 1887, and these were very popular in microscopy. Already in 1882, Paul Ehrlich used urinin, which is the sodium salt of fluorescein, to track secretions in the aqueous humor of the eye. This is the first in vivo use of fluorescence. And I'm sure many of you have the eye exam where they have the little blue light that goes across, and you see green. That's the fluorescein fluorescence. In fact, if you do that, you can say to the ophthalmologist, you know, that looked to me like that was about a 4 nanosecond lifetime. And you will impress him with how accurate you are. The disodium salt of fluorescein was often called urinin because of the intense green fluorescence, which was reminiscent of the color of uranium glass that they had. And in the 1800s, uranium glass had small levels, 2% of uranium added, was being produced in London. They don't make it anymore because it's radioactive, and the fluorescence is from uranium oxide. And these things are still out there today, many of them. They also call them Vaseline glass because of when they're not excited, they have this Vaseline color. But you can go in antique stores and see uranium glass. You can take a Geiger counter and probably track the radioactivity. But what about fluorescence microscopes? Because for modern cell biology, the only reason it became important was development of fluorescent microscopes. And they were already made in 1911 by von Heimstedt, Otto Heimstedt, Richter, and Lehmann made these fluorescent microscopes. And that's what an early fluorescent microscope looked like, Heimstedt's thing in 1911. There's a beautiful history and story of all this. I give the reference for anybody interested on the origins of modern fluorescent microscopy and probes. It's a wonderful book. But the earliest observations of autofluorescence, natural fluorescent molecules in living cells, was made by Hans Stubel using one of those microscopes in 1911. He investigated autofluorescence of bacteria, protozoa, animal organs, and we're going to talk later about how important autofluorescence is in modern clinical chemistry. It's used quite popularly now. And one of my heroes of the early people was Stanislaw von Provozek, was the first to introduce extrinsic fluorophores to study cells in fluorescent microscopy. He initially used acridinine, but he used fluorescein, eosin, natriuret, and quinine. And he has a wonderful paper for the fluorescence of cells in 1914. And the sad thing was he was studying cholera, and he died in 1914 of cholera from studying it, but he managed to publish the paper before he died. But he was a heroic figure, and he was the first to use fluorescent dyes with microscopes. Albert Kunz in 1941 labeled antibodies with fluorescein isothianate, giving birth to the field of immunofluorescence. But it was in 1958 when Riggs et al. Reported fluorescein isothiocyanate, a derivative which is much easier to synthesize and has much more stability. So FITC became the popular probe. And if you look at PubMed, which apparently I did on May 1st, you get 332,660 references to FITC. By far the most common fluorescent probe, and it's still used today. People still use it, you know. It's a wonderful probe. It attaches to amine groups. That's why the isothiocyanate will attach to amine groups in proteins. So you have a protein, you can attach this to lysine residues. Now, Gregory Weber, my advisor, in 1952 synthesized danzo chloride for attachment to proteins to use polarization, which is an important technique, to study protein hydrodynamics. Remember, back then they didn't have crystal structures. The only thing you could do were these hydrodynamic studies to see how they rotated in solution to get an idea of the size and shape. And this started the field of quantitative biological fluorescence. And another important pioneer was Osamu Shimomura, who discovered the green fluorescent protein in the Aquaria Victoria jellyfish, which, as I'll talk in a bit, is very important. In fact, this has made pivotal changes in cell biology, this discovery. He was actually interested in a protein, a quarid, which was a bioluminescent protein, that if something happened to the jellyfish, disturbing it, you would get a wave of calcium that would get the aquarium to give off blue light. So it's not absorbing light, it's bioluminescence from calcium. But that blue light was absorbed by a green fluorescent protein right next to it in the membrane. And then it would basically transduce the blue to green, which, why do you want to do that? Well, that's why the sky is blue, right? Because blue light scatters much more as you go to lower wavelength, it scatters more. But by transducing from blue to green, this flash of green light would go farther in the ocean. Now you would ask the question that I asked of marine biology, why does it do this? Why does it want to give off a flash of green light? And you always hear things like, oh, it's good for fighting mates or scary predators. No, they said no. The reason they agree on now is when it was being attacked by a predator, it would give off a flash of light in the hope of attracting a bigger predator that would then come for. So basically, when any predator saw a flash of the light, they'd go knowing that smaller fish may be attacking it. So this was a self-defense mechanism. But now we do it in the lab with recombinant thing. You can grow tons of it in the lab. But back then, he isolated his first sample from 30,000 specimen of jellyfish. And he got 70 milligrams out of that. And I suspect if the students today had the isolator from jellyfish, you wouldn't see much use of this. The jellyfish weighed 1.5 tons. And in his paper, he described that he had to cut the ring off of this, and he got so tired with scissors, he made this ring-cutting machine so he could cut the rings and isolate it. But I want to talk just a moment about it. So the GFP is this protein, has all these beta barrels, and 3 amino acids come together in the interior to make a fluorescent molecule. There's an auto-oxidation. We won't worry about the chemistry. It makes it fluorescent. Well, now it turns out if you make mutations anywhere in this fluorescent proteins, it can change the color. And you now have it from green, you have blue, red, orange, yellow, green, blue, indigo. So you have all these different color fluorescent proteins, all due to changing an amino acid, which they would just change it and see what happens, change it and see what happens. Largely Roger Sen and his group, and all these guys got Nobel Prizes for this, because that allows you then, here's the whole point, you have your protein of interest, you're a cell biologist, you use molecular biology, you attach the DNA for the GFP to your protein, then in the cell, your protein is made with the GFP or whatever, red FP, any attached. So now you've labeled it in the cell, and you can look with the microscope and see where it is and what it's doing. You see how important that became. And as we'll talk in a minute, the different colors were important to allow you to do energy transfer, which I'll talk about very soon. So, but microscopy really was, fluorescent microscopy was slowed by the fact that the fluorescent probes like fluorescein were not so photostable. When you focus a microscope down to it, it would bleach. But then Haugland started to make these really exceptionally fluorescent photostable conjugates. He called Alexa probes. He named it after his son Alex, okay, Alexa probes, which are very popular today. And now there's a whole series of photostable probes, even better than that. And the most popular are the Genelia Farms probes from the famous Genelia Farms Howard Hughes place by Luke Lavis, who's got the latest award we gave out for this. He's coming next week to Genoa to give a special talk for us. But he made these squarene-derived things which are much more photostable even than Alexa. And the great thing I liked and why I had nominated him for the prize, like Weber, he makes them and he gives them out free. You don't have to buy these. You can write Luke Lavis what you want and he'll send you the probe for free, which is really in the spirit of how things should be, you know. So that's why he got the Weber Prize. But let's go back in history a little bit again. Galileo never worked with fluorescence, but he didn't know it was phosphorescence at the time, because a Bologna shoemaker discovered this stone, which turns out to be barium sulfate with impurities, that if he had it outside, then at night brought it in by the fire, in the dark it would glow. And of course, we now know it's phosphorescence. But Gaviola was so smart, he wrote, and this is translated from his document, it must be explained how it happens that the light is conceived into the stone and given back after some time as in childbirth. So he recognized this time delay, which can be seconds and seconds in phosphorescence, but nanoseconds for fluorescence. But when was it first the fluorescence lifetime measured? The first serious attempt came from R.W. Wood, a famous American physicist, who wrote this paper in 1921, which I have it, of course, I like, the time interval between absorption and emission of light. And his idea for an experiment was so beautiful. It absolutely didn't work,
Okay. Yeah, yeah, yeah. Time for questions. So please go ahead.
Probably not many questions about a history lecture because you don't want to show how old you are. There is a question.
So while we wait for some of the younger people to think of a question, I have actually two. One is, has there been any impact, positive or negative, of the use of artificial neural networks in your field? And the second one would be, are you feeling the also impact of the current American government policies on your research?
I should be very careful what I say
Okay.
because I would be extremely critical. And yes, not only is this collapsing the funding, the NIH budget has been cut in half, and many people are losing their research grants now. Also, even for the Weber meeting, several people could not come because they're afraid to go back in the U.S. with the visa. We give out an award every few years for the best PhD thesis in fluorescence. And a young student who's now a postdoc in Stanford won it. And it involves a $1,000 prize and a trip to the Weber meeting. And he just told us Stanford told him don't go because you'll never get back. On the other hand, that saved me $2,000 so for the travel, but that's been very negative. Artificial intelligence, I haven't seen that yet except in one personal case, which I can relate. When I was doing the second edition of this book, and I was in the wheelchair because I broke my hip, and I sent these figures off, and they sent back, we need 60 of these, don't have high enough resolution. We have to make them high resolution. I said, how do I do that? I was very depressed in my wheelchair at home, and my neighbor, who used to be chairman of microbiology, Rick Deckaway, came over because he would bring my mail and bring me some food, and he was in digital photography as a hobby now. He said, oh, send him the figure. Instantly, he did it. And I got on my walker, and I hobbled over to his apartment with a flash drive. All 60 figures he did in like 15 minutes. I said, how is this possible? He said, well, it's all artificial intelligence. Because you have a figure of low resolution, 800 by 800. You want to make it 3,000 by 3,000. The program calculates what color you need in between, and it generates. They said you're not supposed to use them for primary publication, but I'll tell you, if you're ever doing something like this, improve the resolution of a picture, I cannot tell that it looks perfect, you know. But in terms of broader applications, I know people are trying now to do, even with phasers, I've heard of some people analyzing like many, many phaser things using artificial intelligence, but I myself am ignorant of the, I barely have enough native intelligence to do it. I know nothing about that, but it's having an impact for sure. And certainly the changes in the government are having an impact. So let's hope we can still talk a couple years from now, but who knows what will happen. Any less depressing questions?
More questions. Yeah.
Okay, here we have a group of young students from the high school, and they are starting doing some research. So can you give them some advice about this, about starting research?
Well, I think the important thing at any age, and I still have it, is to enjoy what you're doing and try to do something that interests you. And whatever you're assigned to do, of course you can find something interesting there. But one nice thing with fluorescence, I think, is it's brilliant colors. I always tell people in the lab, they put things in the machine, and I say, look at the color before you put it in to appreciate what's happening. But the important thing of anything with the students, and Lord knows back from my days, I think even now, most of us still have this curiosity. Anytime I hear a term, and it's much easier for you young people now. In my day, when I saw something I didn't understand, I'd go to what we call a library,
Okay. Okay, many thanks for your answer and your holistic approach. Okay, many thanks.
More questions? Okay, thank you.